Rothenberg. Good day, everyone. I'm Marc Rothenberg, the president-elect of the International Eosinophil Society, and it's my great pleasure to welcome you today to our webinar, which is on cross-evolutionary and cross-species eosinophils. Next slide. Next slide, please. As you know, our society normally has meetings every 2 years. However, during the COVID pandemic, we've been reverting to webinars, and we are very pleased to announce that not only will we be continuing the webinars over the next year, but we are now planning a meeting in physical presence in July of 2023, and it'll be hosted by our esteemed colleagues in Hamilton, Canada, and by the McMaster University. And more information will be following that at the end of the webinar today. So please keep that in mind, and we'll be sending out reminders about that meeting. Our society is an organization of scientists and clinicians interested in the eosinophil. The society sponsors biennial meetings to review new information about the eosinophil and its role in health and disease. And the society has been established to bring together scientists around the world who are engaged in research in the field of eosinophil biology, eosinophil disorders, with a view to facilitating exchange and ideas and information about this fascinating cell that's featured on my tie. And some of our speakers, like the next one, will be showing you various forms of eosinophils on his tie. Next slide, please. In terms of logistics today, please use the question and answer button to ask questions anytime during the presentation. All attendees are muted and the chat feature is disabled. If you have technical difficulties, try logging off and logging back into the webinar. The webinar is recorded and will be posted on our website. Next slide, please. It's a great pleasure to moderate the session today. And our first speaker will be Professor Steve Ackerman, who is the professor of biochemistry and molecular genetics and medicine at the University of Illinois at Chicago. His talk today will be on cross-species and cross-evolutionary considerations in eosinophil biology. Dr. Ackerman, please take over.
Thank you for the kind introduction, Marc, and the invitation to present on the cross-species and evolutionary biology of the eosinophil. Oh, sorry, not advancing. Oh, there we go. So before I start, I want you to all take a quiz, and hopefully by the end of this, you'll be able to answer these very important questions. Father of myeloid cell biology, infamous eosinophil ophile or eosinophil philosopher. Eosinophils are unequivocal liars, and of course, on the origin of species by natural selection. The origin and evolution of the eosinophil has received very scant attention since Ehrlich first named it for its eosinophilic staining acidophilic type granules. But there was a recent consideration of evolutionary origins and the presence of eosinophils in extant species, which was a scholarly review by the late Michael McGarry, which was published in the IES 2013 monograph on eosinophils and health and disease. The identification and potential functions of hemocytes in particular, which can show eosinophilic granulation, shown here for Cranwell's horned frog, offers some insights into the origins and functions and roles of eosinophils, both in extant and contemporary species, particularly in processes that involve transformational changes, such as metamorphosis in insects and amphibians. The participation of hemocytes in invertebrates in various aspects of morphogenesis likely prestaged the contemporary functions of eosinophils in vertebrates, particularly in terms of tissue remodeling and homeostasis during development, but also in terms of remodeling and fibrosis in eosinophil-associated diseases in terms of their pathogenesis. Most descriptions of granulated hemocytes show them to be electron dense, not eosin staining, but their granule ultrastructure is suggestive of their being acidophilic. The literature is replete with examples of these granulated hemocytes in a variety of insects, oysters, and clams. And they're present also in annelid roundworms, arthropods, and a variety of other invertebrates. But it's impossible to determine if they actually represent an evolutionary precursor of what we consider to be modern-day vertebrate eosinophils. The links are based on biochemical and genetic similarities, but they're highly limited. But they do include the expression of the family of myeloperoxidases, of which, of course, eosinophil peroxidase, as we know, is highly eosinophil specific, at least in human and mouse. And based on protein sequence alignments and phylogenetics, the myeloperoxidases expressed by eosinophils, EPX, appear to have diverged well before that of mammals about 60 to 70 million years ago. However, it's unfortunate that these types of analyses are really not sufficiently robust to indicate when early invertebrate to vertebrate evolution of the eosinophil lineage occurred. Eosinophil-like cells from invertebrates are represented in the majority of phyla, and they show a fairly high level of conservation of this type of granulocyte, the hemocyte, seen in arthropods here, horseshoe crab, insects, giant cockroach, mollusks, and echinoderms, the sea cucumber, as an example. In terms of early vertebrates, inflammatory reactions in ascidians show phagocytes that have electron-dense cytoplasmic and some acidophilic granules that participate in a variety of types of encapsulation reactions induced by foreign bodies. Vertebrate eosinophils, of course, have been identified fairly extensively in representative species, going from fish all the way to mammals at multiple levels, light, electron microscopic, and biochemical. Presumptive eosinophilic granulocytes, interestingly, with morphologic and functional characteristics that are very similar to mammalian eosinophils, have been described in very early vertebrates. The example here being that of the lamprey eel larvae of the Cyclostomata superclass. And there are more definitive examples of blood eosinophilic granulocytes with similar nuclear and electron-dense granules in terms of their morphology that are present in cyclostomes through the cartilaginous fishes like the dogfish shark shown here. The 5 classes of vertebrata show ubiquitous presence of eosinophils in this subphylum, from including mammals, aves, birds, reptiles, amphibia, and the bony fishes. In terms of evolutionary origins and considerations, it's notable that the coelacanth contains eosinophils. In here, from an ultrastructural study of the blood cells of the coelacanth Latimeria columnae, in which the authors identified multiple types of granuloleucocytes, including neutrophils, eosinophils, and basophils. Notably, the coelacanths are found as fossils in rocks from the end of the Permian to the Jurassic period, 251 to 145 plus million years ago. But the current modern day coelacanth, of course, is not quite a living fossil. It's very interesting to compare the morphology of vertebrate eosinophils, and you can see here a layout from the stingray, 2 different types of stingray, all the way to the non-human primates and human eosinophils. And you can see the considerable conservation of morphology in terms of the eosin staining of the granules, and in many cases, nuclear morphology. If I showed you this cowtail stingray eosinophil, you might not be able to distinguish it from a human eosinophil. Non-mammalian eosinophils are likewise very comparable in their structure in terms of their eosin staining and nuclear morphologies, but notably the eosinophils of reptiles are more variable in how they appear in terms of the numbers of granules, nuclear morphology, and eosin staining. And this can be seen here for reptile eosinophils, comparing, alligator, boa constrictor, crocodile, which looks very much like the horse eosinophil, and others, showing this considerably more diverse appearance of eosinophils from reptiles. So the blood eosinophils of reptiles are notably more diverse in color, size, and density. Their granules, their nuclei are quite variable, single, bilobed, oval, eccentrically located. So there's a lot more morphologic diversity seen within the reptilian species. Cytochemical studies of reptile eosinophils show variability in staining properties, and their ultrastructural features are, however, consistently show electron-dense granules. There's some season and species variability affecting the peripheral blood levels of eosinophils in reptiles. Some, like turtles, have high circulating levels, while others have very low or no eosinophils at all, for instance, healthy snakes. There's no short of descriptions of eosinophils in amphibians, particularly frogs, as they're present both before and after metamorphosis occurs. Typical eosinophil morphology is seen with eosin-positive cytoplasmic granules and ultrastructure. And notably, observations in frogs, especially early ones that I'll come to momentarily, support a role for eosinophils in tissue remodeling aspects of the metamorphosis that occurs during the development of the frog from the tadpole, particularly in the tadpole gut, which is accompanied by significant eosinophil infiltration. Another interesting point in terms of other species, for instance, avian species, is that there's highly identical morphology to mammalian eosinophils, and notably, as I'll touch upon momentarily, in the chicken, the transcriptional regulation of eosinophil development from early myeloid progenitors to the mature cell is very similar to that of humans. And of course, there are peroxidase containing eosinophils that have been identified in both embryonic and adult zebrafish, which provide a potentially useful vertebrate model for their study, and I'll touch upon that momentarily as well. In 1923, as early as 1923, there were observations made that eosinophils may contribute to local tissue remodeling. You can see here a quote from this paper by Jordan and colleagues in JEM, describing many eosinophils near the basement membrane of the epithelial lining. The granulocytes, both special and eosinophilic, are mobile. They have a lytic effect on tissues undergoing regressive change, and they made a point here, whatever initiates the degeneration process, the granulocytes aid in furthering it. So in this very narrative type of publication, there was identification of the eosinophil and its role in early and local tissue remodeling in amphibians. Zebrafish are a peroxidase-containing eosinophil that are potentially useful as a model that can be genetically manipulated for studies of eosinophil development and functions. In this paper, identifying these eosinophils by Bala in David Traver's group in 2010. They identified a high GATA2 high set of cells that they purified and were able to show morphologic, histochemical, and ultrastructural properties, identifying them as eosinophils, shown here, and also negative staining for myeloperoxidase and typical electron-dense granules seen by TEM. In terms of the evolutionary odds and considerations with regard to zebrafish, when zebrafish become infected with the nematode P. tomentosa, this can lead to colonization of gut and peritoneal cavity with increased local tissue eosinophils, and in the gut of the uninfected fish, there's a baseline level of eosinophils in the basal lamina propria, whereas infected fish show increased eosinophils at that site and distillate into the villi, with about a threefold increase in the intestines of infected fish. Mammalian eosinophils, as I said, show considerable beauty and diversity. In particular, you can see the similarities of elephant eosinophils, Asian and African, to that of human eosinophils and primate eosinophils like the lowland gorilla and the chimpanzee, contrasted to those of the horse with the very large eosinophilic granules, which are very comparable looking to that of the crocodile. In mammals, eosinophils are professional liars. And there are numerous descriptions, of course, available for many, many, many species. And of course, as we're going to touch upon in Beth Jacobsen's presentation, multiple wild, hybrid, mutant, and genetically altered mice. In a review published by the late Jamie Lee, he identified eosinophils as regulators of local immunity and or remodeling repair under both healthy and pathologic conditions, formulating the liar hypothesis. Liar makes a very strong case for the primary function of eosinophils being modulators of innate and adaptive immune responses, inducers of remodeling and fibrosis under pathologic conditions of tissue eosinophilia, and regulators of normal tissue and metabolic homeostasis, at least in the mouse. As we're going to hear from Amy Klion, these functions may not be obvious in humans in terms of studies looking at eosinophil deficient humans induced by biologics. In terms of the classically considered role of the eosinophil in host defense against multicellular parasites, the eosinophil, of course, is very well equipped to kill them or to aid in their encapsulation in terms of their eggs and larval stages. Notably, early appearance during evolution and many studies of eosinophil responses to helminths and other parasites in all of the eosinophil deficient mouse strains argues against conferring any significant selective advantage in host defense during eosinophil evolution. And the absence of significant life-threatening developmental abnormalities or functional deficiencies in eosinophil deficient mouse strains listed here, and the now biologic-induced eosinophil deficient human begs the question of why the eosinophil lineage continues to be ubiquitous in vertebrate species. As well, the presence of resident eosinophil populations and subtypes at a homeostatic baseline in bone marrow, mammary gland, GI tract, thymus, and the endometrial lining of the uterus argue against the eosinophil's major function being that of in host defense against pathogens. Another notable point is there's considerable conservation of transcriptional mechanisms that regulate eosinophil-specific gene expression, particularly in eggs or birds. There's conservation of both transcription factors and transcriptional mechanisms that regulate eosinophil-specific gene expression. Eosinophils contain a whole series of regulatory sequences that bind CEBPs, ETS factors, and of course, GATA factors, which are extremely important and required for eosinophil development. And in the chicken, human, and mouse, CBPs repress FOG, which is normally a potent coactivator of GATA1, but in the eosinophil is a potent repressor of GATA1, so it has to be downregulated by CEPs to allow eosinophil-specific gene expression to occur. As well, there's a very highly conserved high affinity double GATA site in the promoter enhancer regions of GATA1 itself, and many other GATA target genes expressed in eosinophils like IL-5 receptor alpha, MBP1, CCR3, and others. And these confer highly eosinophil specific gene expression, and this occurs across vertebrates from zebrafish to humans. Eosinophil associated proteins also speak to conservation of form and function across vertebrate species, and I'll go through some of these. There's the eosinophil major basic protein is expressed in both humans and mice. There's orthologous gene pairs for MBP1 and MBP2. There are significant differences between humans and mice in terms of gene expression and protein biochemistry. In man, MBP is very cationic, but MBP2 has lost this characteristic, but both retain their cationicity in the mouse. So there's a difference between human and mouse. As well, humans MBP is expressed in non-leukocytes such as placental site X cells, which leads to an accumulation of the unprocessed proform during pregnancy. But placental expression and serum accumulation of pro-MBP does not occur in the mouse. Eosinophil-associated ribonucleases, or the EARs, are a very rapidly evolving group of genes. The EDN-ECP genes likely arose from a duplication event that occurred after the divergence of New World and Old World monkeys somewhere around 30 million years ago, as reported by Helen Rothenberg in Nature Genetics. And the accumulation of many non-silent mutations at high rates that exceed those for other functional coding sequences in primates, where while there's a retention of structural and catalytic domains required for ribonuclease activity, suggests that these two genes, EDN and ECP, are responding to unusual evolutionary constraints, perhaps in terms of their physiologic functions, for instance, host defense against viruses. And finally, mouse and other rodent ears are not orthologous to human EDN and ECP. They represent distinct paralogs as opposed to homologs that occurred well after divergence of primates and rodents. Eosinophil peroxidase is another example, expressed both in humans and mice as orthologs, and they encode proteins displaying very significant peroxidatic activity. There's extraordinarily high sequence identity at the amino acid level, greater than 94%. And in both species, EPX is part of a closely linked leukocyte pair of peroxidase genes, myeloperoxidase and eosinophil peroxidase, for which a duplication event occurred again before the divergence of primates and rodents. Among the granulocationic proteins in humans and mice, eosinophil, of course, is the most highly eosinophil specific. Finally, CLC protein, or galectin-10, is found only in primate and not non-primate mammalian species. And despite a very high level of expression in human eosinophils and a conserved galectin gene family in mice, this protein is completely absent from the mouse. So there's no murine ortholog or obvious paralog gene sequences in the mouse. It's also one of the very earliest and most highly expressed genes during eosinophil development. And we recently showed that it's required for granulogenesis in developing eosinophils, playing a role in vesicular transport of the ribonucleases, both during differentiation and piecemeal degranulation. Of course, expression is not limited to eosinophils. There are gene transcripts in other cells like basophils and Tregs. And in Tregs, it appears that CLC Gal10 is required for their immunosuppressive phenotype. And then there are recent reports that galectin-10 promotes allergic immune responses and also mucus plugging in the lung in severe asthma. So to conclude, what are the roles of eosinophils in normal tissue and metabolic homeostasis? I think the major functions include maintenance of tissue microenvironments. I've shown you a lot of data for that, both during normal organismal development, but also as part of host immune responses in allergic reactions and other pathologic conditions. But of course, I think it's important to point out, and we'll cover this in the next two presentations, most of the findings from mouse models have yet to be confirmed in humans. Thank you, and I'll be happy to take questions.
Thank you, Dr. Ackerman. It was a beautiful talk. Absolutely great slides and images. I think nothing tops your tie, though, for images of eosinophils. I see only one question so far, but I encourage others to enter questions into the Q&A box. I wanted to start off by just asking you a question. You've shown some elegant images of eosinophil-like cells that go back quite a long way in evolution. By most work on eosinophils in mice has shown their diverse functions besides just pro-inflammatory roles. But in humans, which we'll hear about later, probably there seems to be less, there seems to be very limited evidence that eosinophils are doing anything beneficial. So is there any evidence for eosinophils having any functional role outside of mice in any of these species that you've talked about, even in zebrafish, where it's relatively easy to knock out eosinophils from morpholinos to see if there's any consequences for eosinophils, particularly under homeostasis?
Yeah, that's a good question. In terms of zebrafish, there haven't been a huge number of papers following up those initial papers identifying eosinophils and showing how they play a role in responses to nematodes, for example. I didn't have time to show the slide, but they also are induced and present if you induce allergic reactions with parasite antigens and allergens. But unfortunately, it's not been a lot done using zebrafish as yet. But you're right, it does provide an opportunity to knock out eosinophils and study their deficiency in the zebrafish. There's a large number of editorials recently. There are a number of editorials that have been put out by Jerry Gleich and Chris Leiferman, for example, arguing that the human experiment where eosinophils are knocked out using biologics that target IL-5 or IL-5 receptor show really no pathology, and I think Amy Klion is going to touch upon this in her presentation, arguing that the role for eosinophils is still an open question in terms of translating the findings that we are hearing about over the last 10 years in mice to humans.
Okay, thank you. We're going to start off with the question by Amy Klion. In the zebrafish model, that it's been able to find anything similar to IL-5. Have IL-5-like molecules been identified in other mammals?
IL-5-like molecules are present in non-human primates and mammals. I'm not sure. I don't remember what the corresponding cytokine is in zebrafish that participates in their development, in their initial development, and also in their priming and activation. I'm sorry, I can't answer that one.
Okay, thanks. We've got a question from David Jackson. He says, great talk. Is there any evidence that eosinophil numbers in the blood tissue are higher in parts of the world with higher parasite exposure?
Okay. Can you repeat the last part of that question?
Basically wants to know if eosinophil levels are higher in parts of the world where there's increased parasite exposure.
Oh, yeah, I think the answer to that question is definitely yes. Yeah.
Mm-hmm.
Blood eosinophil levels tend to be higher in areas where individuals exposed chronically to helminth infections, for example.
Okay. In terms of other questions, I don't see any coming in right now, so I'll ask one more, which is a little bit of a follow-up to my first question, but it's maybe a theme for further discussion. So in the mouse, I mean, there's a lot of elegant experiments, basically manipulation of mice to illustrate different functional roles for eosinophils. But the bottom line is that these mice that are knocked out for eosinophils do breed normally, and they don't have spontaneous cancer, but maybe we'll hear differently, but only have these defects when carefully examined under extreme conditions. So I wonder, and now it may be consistent with the human data that shows no major defects also. But I wonder how much of this is the fact that we've experimentally manipulated the mouse, and then we come to all these other species, because when you see the conservation of eosinophils going back to, like you said, hundreds of millions of years, it really does suggest that there's a physiological benefit to having these cells. Maybe it's about host defense, but there may be other roles as well. So this is a question or maybe a comment that we could bring out forth as the other speakers continue. There is another question that emerged from Jerry Gleich. So if you have any comment about what I said, just please keep it very short so we can make sure to bring up
I can take
Dr.
Jerry's
Gleich's
question. How about I take Jerry's question?
Okay, you'll take Jerry's question. Are eosinophils important in snake skin shedding?
Good question. I don't know the answer to that. I don't think anybody studied it.
Yeah.
That's for
kind
your next
of a
career,
follow-up to
Jerry.
what I'm saying is, are there any physiological roles for eosinophils in any other species that have been identified? And skin shedding, maybe he can explain why he's focused on that, but that would be interesting to know.
Yeah, it would be. It's an interesting question, but I haven't come across anything about that. And there's
gonna
really
move on
scant
now.
information on reptiles in terms of eosinophil function.
Well, thanks a lot for this illuminating visual talk and great images and thought for discussion to lead into the next talk. Next slide, please. Introduction to the speaker, Dr. Amy Klion. who is well known to the eosinophil field. She is the chief of the Human Eosinophil Section Laboratory at Parasite Diseases at the National Institute of Allergy and Infectious Disease. Dr. Klion, we're looking forward to your talk.
So thanks very much for inviting me to talk. And Steve, thank you for the perfect introduction. All the slides that I eliminated were in your talk. So can you, oh, sorry, that's the last slide instead of the first slide. So can you see my presentation? Yes, no?
Yes, we can see it. Thank you.
Okay, great. So I changed my title. The title in the email was Eosinophil Depletion in Humans, an Update. But I made a tactical decision that talking about eosinophil depletion and the success in treating eosinophilic disorders really would be the topic of an all-day symposium. There are lots of agents that are currently available, as I'm sure everybody knows, and their effects in different diseases is really quite different. So I decided instead, and this is why I said Steve gave me the perfect introduction, to focus on eosinophil depletion in humans. Is it safe? So just to sum up some of the things that have been reported in terms of mouse data on regulatory roles, as Steve mentioned, the liar hypothesis was really put forth by Jamie Lee in 2010. But subsequent to that, there were a number of reports of roles of eosinophils in mice as homeostatic roles and roles in specific situations. This included maintenance of plasma cells, glucose metabolism, suppression of type 1 gut inflammation and induction of Tregs, anti-tumor activity, improved cardiac function post-MI, and a wide variety of other things. And what I'm going to try and do in the context of this talk is address these things in the context of what we know about humans in the setting of eosinophil depleting therapies. Unfortunately, and Jerry Gleich and Steve and I put together a manuscript a number of years ago, there really aren't, there's almost no data on natural absence of eosinophils in humans. And this is in part because 0 is a normal eosinophil count in most laboratories. 0, it may not really be 0 if you count enough cells, but we don't really have the capacity to look at large data sets and say, well, these people have no eosinophils. There are rare conditions associated with absence of eosinophils, but that's usually in association with a number of other findings, as in Good syndrome, where there's a thymoma. And so it's really not very clear. And I think the advent of biologics and other targeted therapies is really providing us with the ability to answer these questions in humans. So the first thing we have to consider is, what are the therapies that are available for eosinophil depletion? And this is just a cartoon showing some of the drugs that are currently available to make the point that there are very different acting drugs. So mepolizumab and reslizumab bind IL-5, and IL-5, as we know, is really crucial for eosinophil development and maintenance. Benralizumab and lirentelimab, which is an antibody to IL-5 receptor and Siglec-8, respectively, they induce antibody-dependent cell cytotoxicity and deplete eosinophils. There are drugs that interfere with eosinophil trafficking. Listed here are bertilimab and dupilumab, but there are many others that interfere with eosinophil trafficking to the tissues. And then there are drugs that are in development that have other or unknown mechanisms, including antibody to galectin-10, which Steve introduced, and dexpramipexole. Each of these has slightly different effects in the different compartments that eosinophils exist. And this is important when you then try to look at data about safety with depletion. So mepolizumab, and although there's not as much data, reslizumab cause maturational arrest in the bone marrow. They reduce tissue eosinophils, but not completely. And And that's shown here in an old study from Menzies-Gow, where they looked in the bone marrow as well as the bronchial mucosa, and were able to show a decrease, although not a complete elimination, of eosinophil myelocytes, metamyelocytes, and mature eosinophils in the bone marrow, but no change in early precursors, consistent with maturational arrest. This is from a paper by Stein and Marc Rothenberg's group looking at mepolizumab for the depletion of eosinophils and eosinophilic esophagitis. The top is pre-treatment, and the bottom with the red arrows shows that although there's a really dramatic decrease in eosinophils, there are still eosinophils that remain. And the third example is from a paper by Kelly et al. In 2017, where they gave a bronchial challenge with antigen and were able to show that mepolizumab diminished the eosinophil response in the lung, but didn't completely ablate it. And so I think that this is important to recognize, because the case has been made that even though mepolizumab depletes or reduces eosinophils, they're not completely gone. And if they have a homeostatic role, that may persist despite this. Benralizumab, on the other hand, completely depletes eosinophils and eosinophil precursors in the bone marrow and every tissue that's been examined. It also reduces basophils. On the left, you can see data from a study that we did, a phase 2 study of benralizumab in patients. And you can see on the left in the bone marrow, there were tons of eosinophils with their red granules. On the right, there are really none, and there are no eosinophil precursors. This was also documented by flow cytometry. And on the right, you can see the stomach and ascending colon from patients in that study at baseline in week 24, where we were not only unable to show eosinophils at week 24 by H&E, but even very sensitive staining with antibody to EPX showed that there was complete depletion of eosinophils and eosinophil granules at week 24. Interestingly, benralizumab, despite its effect on eosinophils and basophils, has really no effect on mast cells, and this has been documented by others as well. What about the other eosinophil targeted agents? As I mentioned, lirentelimab depletes eosinophils in the blood and tissue through a mechanism similar to benralizumab, but it also, and that's shown in the figure over in the right from a study by Evan Dellin et al. Looking at eosinophilic gastritis and duodenitis, where you can see that the eosinophils are really dramatically depleted in the tissue. It also inhibits mast cell degranulation, and I'll come back to that later. Dexpramipexole, which is a small molecule oral drug that we did a small study with in hypereosinophilic syndrome and has subsequently been studied in asthma, depletes eosinophils in the blood, bone marrow, and tissue nearly completely, as very similar to benralizumab, but it shows a maturational arrest at the promyelocyte stage, which is different than what we saw with benralizumab. So you can see that on the right here with pretreatment and post-treatment bone marrow showing depletion of mature eosinophils, but maintenance of eosinophil promyelocytes shown in the inset. In terms of other tissues, shown on the right are biopsy samples from a patient that participated in our dexpramipexole study. And what you can see is on the left, pre-treatment, on the right, post-treatment. And similar to what was seen with benralizumab, there really is complete depletion of eosinophils and improvement in the tissue. Dupilumab, on the other hand, reduces tissue eosinophilia. This is, as many of you know, an antibody to IL-4 receptor that blocks IL-4 and IL-13 function, but has no effect on eosinophilipoiesis in the bone marrow. The reduction in tissue eosinophilia and improvement in tissue pathology is shown over on the right in a paper by Hirano et al., part of the phase 2 study in eosinophilic esophagitis. Because it blocks tissue transit of eosinophils, it actually can cause a transient increase in blood eosinophils, which is different than the other drugs described. So I think my point here is that you really have to think about the varied effect of the targeted agent that you're looking at when you start to think about what toxicity might be there. There's some other challenges in identifying safety issues. As Steve brought up, animal data may not accurately predict human outcomes for a variety of reasons. Clinical trials where these things are looked at very carefully are time limited. There are very few studies that extend beyond 1 or 2 years. Post-marketing data, on the other hand, is really incomplete and relies on provider recognition of a potential relationship between the drug and the effect. And finally, adverse effects may affect only selected populations or require specific circumstances. Examples that I'll mention briefly later are helminth-infected populations or the setting of a COVID-19 pandemic. So I'm now going to move on and talk about some of the things that were seen in mice and whether we have any evidence that these things are a problem in humans. I'm going to start with the relationship between eosinophils and plasma cells. So as Steve and I both mentioned briefly, so there is really substantial mouse data for many of these things. And in terms of the relationship between eosinophils and plasma cells, there's very convincing data showing that eosinophils in the bone marrow make molecules like APRIL that help sustain long-lived plasma cells in the bone marrow and the tissues. In humans, the same group showed that there was co-localization of eosinophils expressing APRIL and plasma cells in colonic biopsies of humans, and that's what's shown here. And another group was able to show that there was an association between increased activation and CCR4 expression on eosinophils in the bone marrow in patients with multiple myeloma. So these data suggest, but don't prove, that there's a relationship in humans between eosinophils and plasma cells. I will say that routinely when we examine bone marrows in patients with all types of hypereosinophilic syndromes, when we see increased eosinophils in the bone marrow, we typically do see a mild to moderate increase in plasma cells. However, the big question is, does that matter? One more piece of data, and then I'll get to whether it matters. So in our benralizumab study, we did enumerate plasma cells, and neither plasma cells nor B cell numbers were affected by monthly benralizumab therapy at 12 weeks as compared to placebo. And that data was not published, but is shown here for plasma cells on the left, baseline versus benra by flow cytometry, and then on the right for mature B cells and precursor B cells. And you can see that there's no difference between the baseline and the benralizumab data. We've looked further out than this, and there continues to be no change in patients on long-term benralizumab. So does it matter? In the mice, depletion of or mouse deficient in eosinophils were shown to have abnormal vaccine responses. These were only in response to recall T-cell dependent antigens. And so there's been almost no data looking at this in humans. I'll show you what data there is. There was one study by Zeitlin et al. In patients with asthma, where they gave seasonal flu vaccine after 8 weeks of benralizumab and showed that the responses were comparable to placebo in 103 patients who were only receiving inhaled corticosteroids, and that data is shown here. In our phase 2 study of benralizumab, once patients were on open label benralizumab at 17 to 20 weeks, we gave tetanus booster to patients that had not had a Tdap in the last 5 years. And what you can see is that all but one patient had a normal response to the Tdap booster. It turns out that the one patient who did not have a normal response has combined variable immunodeficiency, and that she had predating her benralizumab. And so she would not be expected to have a response. So a nice control. Finally, one of the patients in our phase 2 study became pregnant. She became pregnant while on benralizumab with no eosinophils at all. So the baby was conceived, grew, and was born without any eosinophils at all. As you can see in the figure here, the baby did not develop eosinophils until 7 months after birth, but had normal pneumococcal and tetanus vaccine responses at 1 year. And so I think all of this together with the primate data suggests that depleting eosinophils in humans and non-human primates really has no effect on vaccination. What about glucose metabolism? Again, this has been shown. There's a lot of data on eosinophils in adipose and eosinophil involvement in glucose metabolism. There's a little bit of data in humans. There's one study that showed increased eosinophils in blood and adipose tissue of patients with metabolic syndrome, and that's shown here, body mass index. I take it back. This is the wrong figure, but this is showing that the IL-16 content of eosinophils was related to body mass index. We looked at this in our benralizumab study. We looked both at weight and at hemoglobin A1C levels, and they did not change before or after benralizumab. We followed weight out to 2 years. And again, there's really been no significant change in weight. The one patient who lost weight was on quite high doses of steroids when he started the study and has lost weight as he's come off steroids. Eosinophils and GI homeostasis. This has been a huge area of interest. Hopefully, Beth Jacobsen will talk about that in her talk. Again, very little data in humans and mostly circumstantial. In one study by Lingblom et al., they were able to show that blood eosinophils from patients with eosinophilic esophagitis have an impaired ability to suppress T-cell proliferation in a mixed lymphocyte reaction. The data from that is shown here on the right. The upper left panel is T-cell proliferation in patients with in eosinophils from patients with EOE or healthy controls, and you can see that there's no difference. Suppressive activity is shown in the upper right panel, and then cytokine production in the bottom 3. And again, there's no difference between the normal patients and the EOE patients. There's a very interesting study in 2016 showing that microbiota in mice could cause release of IL-25, maintaining epithelial integrity during Clostridia infection that was dependent on eosinophils. They did show some human data in this study, which is shown here at the bottom, and confirmed that there were eosinophils in the setting of Clostridia infection in humans, and that IL-5 levels, IL-25 levels, were also increased. Again, really circumstantial information. There's been a lot of interest in looking at this in clinical trials, but despite multiple clinical trials of many different eosinophil depleting biologics in eosinophilic GI disease, worsening of type 1 inflammation, either homeostatic or in response to infection, and or decreased epithelial integrity have not been reported. In a very recent study by Kwong et al. From my group, we were able to show that benralizumab doesn't improve some of the epithelial changes, and that's shown here on the left at baseline, week 24 and 1 year in small bowel and stomach, and then over on the right in the colon. Eosinophils are removed, but many of the other histopathologic changes remain, but it didn't worsen. It just didn't get better. And so I think this is really a topic of interest that needs to be looked at more carefully. Everybody's worried about eosinophils in cancer. The data in mouse models and in humans is really confusing. This is just one example of a paper looking at various types of cancer and whether eosinophils are good or bad. You could pick any type of cancer. You could look at just lymphomas. No matter what you look at, some models and some humans, eosinophils seem good, some they seem bad. Unclear, really unclear what this means. There have been very few, as I said, very long-term studies looking at eosinophil depletion in cancer. This is an example again from Fei-Lai Kwong and my group, where we looked at our long-term patients on mepolizumab. Mepolizumab's been available for study since 2000. And so we have patients that have been on this drug for a very long period of time in this study, up to 11 years. And we found no increase or difference in malignancy rates. I realize that it says 10.9% in control and 17.4% in HES, but this was not significantly different. And there was no predominant type of cancer that was more prevalent in the mepolizumab group. No excess incidence of cancer has been reported in any of the other long-term trials of eosinophil depleting therapies to date. But again, this relies on very few trials and whether or not cancers are reported as related to eosinophil depletion to the FDA. So I think this is still a little bit of concern. Are there other outcomes that require further study? Helminth infection is a big one. So eosinophils, everybody thinks of Helminth infection when they think of eosinophils. In fact, the data in mice regarding the role of eosinophils in Helminth infection really depends on the model, the host, and the parasite. In terms of whether depleting eosinophils has any effect in humans, Helminth infected patients have been systematically excluded from the clinical trials of eosinophil depletion biologics, and studies haven't been performed in areas where exposure to helminths is a problem. And so I think we really don't have data. We did perform, and I'm not showing you the data, but it's published, one study where we administered on purpose reslizumab, anti-IL-5, to a small group of patients with Loa infection to see if we could prevent the eosinophil-mediated post-treatment side effects. It was successful in blunting the eosinophil response, had no effect on the post-treatment side effects, and did not cause any change in parasite numbers prior to treatment or any evidence that there was an effect, at least in that model. But there's really very little information. Autoimmunity, there's been data from murine models in rheumatoid arthritis, as well as liver toxicity and other things, looking for a protective role for eosinophils. Although the data, I think, is really convincing in the murine model, the reported data suggesting that mepolizumab exacerbates rheumatoid arthritis in humans is confounded by the fact that the patients were changing their background therapy and insufficient clinical data is provided. Anecdotally, we have not seen, we follow quite a few patients with rheumatoid arthritis and hypereosinophilic syndrome that have been started on biologic therapies, and we have not seen exacerbation of rheumatoid arthritis in those patients. We have one patient who was on our benralizumab study with ankylosing spondylitis. That patient came off his TNF inhibitor during the trial and had worsening of his ankylosing spondylitis and is now on a TNF inhibitor and benralizumab and doing very well, back to his baseline. Increased incidence or worsening of autoimmune disorders has not been reported in the setting of the clinical trials, but again, these have been relatively short and rely on people noticing those changes. And then finally, and maybe most interesting, I think COVID infection has really changed my perception of what eosinophils might do and what situations they might be involved in. Unfortunately, most of the studies have been very small and uncontrolled using eosinophil depleting biologics, although a few in asthma have suggested worse outcomes in patients on biologics who develop COVID. The largest study to date, which was a retrospective review of 71,000 records from patients with asthma, of which more than 1,000 had COVID and more than 800 were on biologics, really showed no difference. All of the increased toxicity in this study was related to the severity of the asthma at baseline. So I think there are things that really need to be looked at that require further study. There are some other unresolved questions. One of the reasons we may not be seeing problems is that there's redundancy in the immune system that is more predominant in humans, that overcomes the effects of eosinophil depletion in humans. And I think this has implications as more and more treatments become available and are administered simultaneously. I don't think we know what happens if more than one cell or pathway is targeted. There are some drugs like lirentelimab that affect two lineages, mast cells and eosinophils. I think the data, the long-term data from those studies will be very interesting. And then there's increasing use of dual biologics, benralizumab to block the eosinophils together with dupilumab to block the itching in atopic dermatitis. And I think we really need to follow those studies carefully to try to understand what depletion of eosinophils does and whether it just requires other cells to fill in. And I think the biggest problem is we don't have a way to assess and monitor the effects of eosinophil depletion in humans in a standardized way. On the other hand, the benefits of treatment have to be weighed against the potential risks, and all the therapies we use for eosinophilia, including steroids, cytotoxic, and immunomodulatory therapies, have risks. This is a picture of a patient who was in our benralizumab study who had extremely severe skin involvement, was hospitalized monthly because of infectious complications of her skin disease. And on the right is her on benralizumab. If you had asked her, if you had told her that there was a 5% increased risk of tumors being on benralizumab, she would have said, so what? So I really think that we need to think always at the patient level. So I'm going to end there and just wanted to acknowledge my entire human eosinophil section, past and present, especially Fei Lai Kwong, Penise Corey, Fanny Legrand, and Michelle Makia, who've been involved in many of the clinical studies, our clinical parasitology group, industry partners, and my collaborators with whom these studies would not be possible. Thanks very much. Happy to take questions.
Thank you, Dr. Klion. There's a couple of questions from the group. One is, are there any common side effects among all these drugs which could indicate an eosinophil beneficial role?
Not so to date, no. I mean, I think one of the interesting examples was the initial reports with mepolizumab of increased zoster infection. That was not, and so that was attributed to eosinophil depletion, but benralizumab, which depletes eosinophils even better, there was no association. So there's been a lot of discordance and nothing common has been reported across.
Thank you. There's a question from Thomas Ahrens, and he says, great talk. I might have missed it, but is there anything known about the efficiency of eosinophil depletion by different biologics in the small intestine?
Yeah, there's some. I mean, so mepolizumab definitely does not completely deplete in any tissue. So most studies have shown mepolizumab and reslizumab, most studies have shown IL-5 agents deplete about anywhere from 50 to 60%, whereas benralizumab completely depletes, lirentelimab completely depletes. I'm not familiar with the data from dupilumab. I believe it doesn't completely deplete, but Marc Rothenberg, Marc, you may know better than I do about that one.
90% reduction in the esophagus.
Right, but is anything known about small intestine? No.
No.
Not yet. It will be, but not yet.
Right. Thank you. A question from Francesca Levy-Schaefer. I really have two comments. First was the lecture was great, and the second one is not shown here, Francesca. Sorry, but please add that. Thanks for the positive feedback, though.
Right.
Do you have any data about depletion of eosinophils during treatment with immune checkpoints?
No, so I think that that's a very interesting question, and we've been asked about a couple of patients with eosinophilia during treatment with immune checkpoint inhibitors, but as far as I'm aware, there have been no case reports of that in the literature. I routinely check. I haven't seen anything. There could be. Okay.
Francesca's second question is that, as for eosinophils and also for mast cells, it seems that there's no human born in the absence of these cells, and that the first attempt to mast cell depletion does not harm the patients. I suppose the question is to comment about that.
Yeah, no, I think that that's true. It's, again, I mean, mast cells are the kind of thing that it's very difficult to assess. I mean, if you really want to know that somebody has no mast cells, you have to look in the tissue. And there are not, as far as I'm aware, maybe Francesca knows, there aren't really drugs like benralizumab that completely deplete mast cells in the bone marrow. And so I don't know if that, I don't know, it may just be that we're not able to know.
Okay, thanks. Cheryl Gleich has a question. Do you have any comments about the anti-TSLP agents and eosinophils alone or with other agents? I suppose the question is, what does the anti-TSLP do to eosinophils?
So most of the, right, so most of the studies have, and I didn't show this data because I didn't show efficacy data, but most of the, even the anti-IgE, anti-IL-4, IL-13 alone drugs, anti-TSLP, all these drugs in general reduce eosinophils to some degree in the blood. The exception is really dupilumab, and dupilumab also decreases blood eosinophilia in many people. It's just that it has this subgroup of patients where it goes up. Most of the data I've seen with anti-TSLP in the higher level blockades is that they reduce blood eosinophils, and there's not very much data yet. There's no data in bone marrow that I'm aware of, and there's not very much data in tissue yet. There will be soon from the asthma literature.
Question from David Jackson. The dupilumab trial in pediatric asthma showed no helminth infections in the placebo arm, but 7 infections in the dupilumab arm. At the same time, work by Vorager and others have shown a signal for IL-4 and 13 deficient mice rather than eosinophil deficient mice. Do you think we should be more concerned with the broader action of dupilumab than with the mepo-benra in relation to anti-helminth immunity?
So that's a really interesting question. And my gut feeling is yes, the data on IL-4 and IL-13 and helminth infection is much stronger than the data on eosinophil, the importance of eosinophils. I'm particularly worried, as I said, about the possibility of patients receiving both dupilumab and benralizumab, for example. Most of the time, the increased risk of infection is not a big deal. The problem, the only parasite for which there's a potential problem would be strongyloides, and IgE is known to be very important in that setting. So I think this is something that really needs to be looked at prospectively in larger groups.
Okay, thanks. There's a comment by Aline Bosick. She says, well, as the one reporting the influence in rheumatoid arthritis, I'd like to point out that the patients were in remission since months before being exposed to anti-IL-5 therapy, and that the reaction was quite rapid. We think that the long-standing remission phase is an important point. Nevertheless, you are right. A study of a large cohort would be necessary.
So thanks for that. I mean, I only have access to what's published, and the data that's published really doesn't provide any data on steroid use or other things going on in the background or the clinical information about the patients. I think that this really, because the data in mice is so convincing, I really think this needs to be looked at in a closer way.
Thanks, Amy. One final question from me. You talked about eosinophil depletion in humans is not associated with an increase in type 1 immunity. But my question is what about type 2 immunity? Because there's recent data and report that mepolizumab decreases tissue eosinophils, but increases type 2 cytokines in chronic rhinosinusitis by Harvey's group. And in fact, we've seen that as well with benralizumab. We see a paradoxical increase in type 2 cytokines, especially eotaxin 1. So any comments about that?
Yeah, we've seen the same thing. We haven't published that data, but we've seen the same thing. I agree. I don't know what it means. I think it's real, but whether it's just changes in the amounts of these molecules, whether it has any actual clinical implications, I don't know. I think that's the problem with a lot of these findings, that there are a lot of things that have been described, similar to the circumstantial data that I showed for plasma cells. There's a lot of things that you can see, but whether they have true functional implications is really unclear. And it may be that other things that we're not looking at are downregulated or altered in response to those changes that prevent there from being clinical manifestations. But only if we look really carefully and we think outside the box at all the different things that may be interacting are we going to get an answer to this question.
That's terrific. Thanks for another stimulating talk. The final talk today is going to be by Beth Jacobsen, who's an associate consultant in the Division of Allergy, Asthma, and Clinical Immunology in the Department of Immunology at the Mayo Clinic, where she also holds an assistant professorship. Her talk today will be considerations of eosinophil biology and functions in mouse models of human disease and health. Beth, thank you.
Hi, thanks. I'm going to try the share screen. And then, shoot, how do I get my arrow? Oop, okay. So I have to say, the two talks before me were really amazing. And so I almost feel like I don't need to actually present anything anymore. They were so good. Anyway, so this is my presentation on basically what I'm doing here. I wonder how I get rid of this part of my screen. Sorry, it's just bothering me right here. Oh well. My goal here really is to look at mouse and humans. And I just thought this was kind of funny. You know, we're looking at it from the human perspective, we're looking at it from the mouse perspective, and just what can we learn about comparing these two? I'm going to call them, they're both model organisms. So as we know, eosinophils, as shown by Jerry Gleich back in 1998, he obtained autopsies from normal people and looked at MBP staining in those autopsies and showed that eosinophils were found in a lot of different places. So the thymus was interesting, and that's been studied further, lymph nodes, spleen, and the GI tract, that sort of thing. So if you look at human and mouse overall after the last few years, where people have looked at homeostatic conditions in both, there seems to be a similarity of where they're found, where constitutively in both human and mouse, they're found in the thymus, the GI tract from stomach to colon, bone marrow, secondary lymphatics, adipose tissue, and it appears during cyclical or developmental stages. There may be a role for eosinophils in the uterus, ovary, mammary development, and lung, and in disease, there's a wider distribution. So they're also found in the same places in diseases in general. So as we've talked about before and presented before, you can find eosinophils in human and mouse and asthma, helminth infection, GI diseases, a variety of infections, a variety of cancers, transplant rejection, skin diseases such as dermatitis, chronic rhinosinusitis, autoimmunity, and organ injury. And actually, I find organ injury to be one of the more interesting developments for the role of eosinophils. So what are they doing in all these places? They could have good functions, which is, you know, maybe they're helpful for metabolism, maybe they're helpful for microbiome, liver or kidney repair, cancer killing, and maybe they're not so good, as we've seen for asthma and dermatitis and cardiovascular function. So, you know, why do we think that there are good and bad roles? You know, some of it's from associations with humans, human disease and their location in that human disease, but a lot of it also is in mouse models. And, you know, one of those things we want to ask is, why has it been so hard to describe eosinophils in human? And many of those that work with eosinophils know that they are incredibly finicky cells. They're just difficult. They don't like to be handled. They can degranulate. RNA-seq is difficult to do from tissue-derived eosinophils, and imaging with fish has been problematic. So you can't go in as easily to look at the transcriptome of those cells, and then doing proteomics, you're kind of guessing, you know, what should you look at there. They're also difficult to transfect. So you can't really just grab an eosinophil, mature eosinophil, and do CRISPR on it, or harder to do siRNA infection and that sort, or introduction and that sort of thing. So what has happened is, in, you know, before the development of these eosinophil deficient strains, Phil and the Gada strain, there was quite a bit of work looking at the role of IL-5, IL-5 knockout mice, IL-5 transgenic mice. And so that actually led to the biologics that target IL-5. But during that same time that those were being tested, two papers were published in the same Science magazine in 2004, and one was by Jamie Lee, where he described Phil, and I was trying to find the cartoon that he has. It's really funny about how he came up with the name Phil. He basically removed the eos from eosinophils, so they're Phil mice. And so that showed that eosinophils do have a role in asthma, in the asthma model of that era, which was an ovalbumin model. And then there's the double GATA1 mouse, which is a mutation in the GATA1 transcription factor at the binding site for that to be transcribed in eosinophils. That mouse actually was generated a bit earlier by another lab, but here it was used to look at the role of eosinophils, again, in allergic airways. And it was found that the deficiency of eosinophils did have an effect. And I actually think they should have had a picture of an eosinophil at the front of this magazine. So I added this actually in the middle of Dr. Ackerman talking, because I realized I had taken it out, but I'll just add it back here. So novel strains of mice. It's really bugging me. I can't see the top of my screen. So novel strains of mouse, no mouse or human is perfect. And if you go to the review article, reviews in immunology, I do go through a list of the different strains of mice that have been developed and the advantages of these mice and the complications of these mice. Why is it not going forward? Okay. So for example, double GATA1 mice, as I said before, GATA1 is critical for eosinophil development, but there are issues because it is a transcription factor. It can have other effects. So it does have an impairment in basophil IL-4 production. It has impairment in mast cell IgE activation. Red blood cells and hematocrit may be altered, and Helene Rothenberg showed that ex vivo eosinophils maybe are produced in certain situations. The other strain that our lab developed, the Phil mouse, it's a transgenic strain that expresses diphtheria toxin A behind the EPX promoter. And because it is transgenic, it is inserted into an unknown site, and it has an unknown copy number. And as a consequence, these mice are a little bit odd. They have less fur, and only males can be bred. More recently, with the development of EOCRE, so that's EPX controlled expression of CRE, we are now able to actually have a separate model of depleting eosinophils if we cross it to DTR or DTA, so diphtheria toxin receptor or diphtheria toxin A, or we can cross it to one of many, I see a chat. Oh yeah. We can cross it to one of many flox genes, and I think to date there's probably been 24, 25 different genes that EOCRE has been crossed to successfully and published. And the other strain is the conditional eosinophil depletion model. This strain has a few more limitations in that only the immature eosinophils are targeted because of the EPX expression being highest in the bone marrow. So this is almost more a model of you either deplete the eosinophils before your disease condition, or you look at turnover of eosinophils in tissue, but it's still useful. Okay, so here are some basics on mouse and human eosinophil biology. Some of you probably already know this, a lot of this material, but I figured I should go over it in case some of it's missed. I am going to add the caveat that this is being provided with opinions and insufficient knowledge-based evidence commentary by me for your enjoyment. It is a very hard topic to cover in 20 minutes. I'd also like to point you to, I think is one of the last really thorough review articles of human versus mouse eosinophils. It is from 2012. It's from the Lee Laboratory. As you can see, it's also with a lot of colleagues, and it was quite extensive. It's 9 tables of extensive data. So please do review this, but do keep in mind that it's more than 10 years old, and there were, and I was involved, so it's probably not completely accurate. Just kidding. It's helpful, but do update it yourself. So let's look at mouse versus humans for their eosinophil hematopoiesis. So this figure here was actually, this was done by Mike McGarry with Jamie Lee. And he's a trained hematologist. And so he looked at eosinophils in the bone marrow based on their H&E staining and their morphology, and described them as precursor, middle, class 2 and 3, or 4. I actually think this is an art that's not appreciated as much as it used to be, to look at eosinophil stages of differentiation. And so here it's a mononuclear cell, it becomes a circular cell, and then a figure 8 cell where you can start to see the eosin staining. If you look at human, they also start as mononuclear cells, and they do develop into polymorphonuclear cells, although they are bilobed compared to the figure 8 of a mouse. And then there's this recent article by Bonjour and Frontiers, and they have some beautiful electron microscopy images, a precursor all the way to mature eosinophils. And you can see here the nucleus structure, as I described before, the starting of the granules, and they start to develop here. And then here you can actually see the dark granule spots. It's a really pretty nice diagram, but this paper also has gorgeous images of mitochondrial development in eosinophils and during activation. So I just wanted to highlight it for that purpose as well. Nowadays, most people look at eosinophil differentiation in the bone marrow by cell surface markers. So IL-5 receptor alpha, the appearance of Siglec F, and then the appearance of CCR3. Ariel Munitz's group published a really fantastic review article, and within it is this great diagram. This is actually half of it, that shows the transcription factors that are present and important during development, as well as the cell surface molecules that start to appear during the stages of development. And as noted before, human and mouse do separate as far as their mouse goes from CMP to granulocyte progenitor to eosinophil, and human goes from CMP to eosinophil progenitor. So there's a slight variation there. It doesn't seem to, from what I can tell, cause a big change in really how to look at the cells. And they both rely on IL-5 for development. And this is I didn't go into mechanisms of how we actually differentiate the cells. I had it in here and took it out. But they both rely on IL-5 in order to mature. So mature eosinophils, they look similar, yet they're different. So in mouse, if you look at a resting or blood eosinophil, it's got this circular shape. There's a lot of pictures out there of in vitro activation or lung activated eosinophils. Even bone marrow derived eosinophils tend to have this phenotype, which is this polymorphonuclear structure. And honestly, if you took out the eosin stain, you might think that's a neutrophil, which might, sometimes can lead to confusion about what cells people are looking at with H&E stain. And then here again is an allergen model where eosinophils have been shown to have also these vacuoles present, and which is a sign of activation. Human eosinophils are bilobed, and they don't tend to look too exciting when they're activated compared to this change in nuclear structure, but they do become hypodense because of their degranulation as they become activated. So here's an incomplete list of ways to identify eosinophils by flow cytometry, because this is primarily how this is done now. And the one thing to consider is that a lot of these change in expression level as far as the intensity, the mean fluorescence intensity, the amount of production, depending on their activation state or if they're in tissue. So both mouse and human eosinophils express IL-5 receptor alpha and CCR3. Those are the classic markers for those cells. I will say that both of those can be downregulated significantly once they're inflamed in inflamed tissue. Mouse eosinophils express Siglec F, and this changes again based on activation state. Humans, they express Siglec 8, and I actually can't remember if that's changed based on activation state. I do not think it is. F480, which is considered a macrophage marker by most, is actually expressed at a medium level in eosinophils. Sort of the equivalent is the EMR1 on human eosinophils. And then you get into markers that are somewhat species specific. So GR1, which is not expressed on human, is important on mouse. And I'll give an example of Ly6G expression on mouse eosinophils. Whereas in human, you look at CD14 and CD16, you're looking for them to be negative for these, although this can vary. CD11B is expressed on mouse eosinophils, but again, the intensity can change based on this activation state. And CD11C is also tissue expression dependent, and Lisa Spencer is the expert on CD11C in gut eosinophils. So the next question is, what about their activation states. So CD63, it can be used for both mouse and human eosinophils. It is a sign of the crystalloid or the specific granule being at the surface. CD107A, I have seen used more recently, although honestly, this has not fully been proven as a sign of degranulation in eosinophils. It is assumed, it seems, from the literature that is a degranulation marker from NK and T cells. So it'd be nice if we actually get in and get some EM images with a CD107A. CD101 is conflicting. I have seen this in regulatory eosinophils. I've seen it in resting eosinophils. I actually think it might be somewhat tissue dependent as well. CD69 used to be a marker of degranulation back when I started a long time ago. Now we realize it's more a sign of tissue, a localization, activation, not degranulation. But tons more on this. Gotta make sure. Tons more to go over here, way too much for me to cover. I have listed quite a lot of reviews that are excellent on this topic. So I can't read the top of the screen. Oh yeah, okay. So one thing to keep in mind is the difference between eosinophil and neutrophil markers that is starting to evolve. So Mesnil and colleagues showed that neutrophils in mice can express IL-5 receptor alpha. Very recently, it has been shown that human neutrophils also may express IL-5 receptor alpha. The function and role of this is entirely unknown. And Sergei Berdnikovs has written a review article that goes into detail about somewhat the plasticity of development of eosinophils and how there might be evidence of expression of some of these neutrophil type markers. And what does it mean for, is it truly a neutrophil or eosinophil? I recommend reading this. And this is really confusing because, you know, are you looking at eosinophil? Are you looking at a neutrophil? Because there's a really interesting publication from Mayer that showed the mice on the Isle of May have expression of Lys6G, which used to be this gold standard for neutrophil markers. Previously, Helene Rosenberg's group had shown with a fungus model a tiny population of Lys6G, and no one had really fully confirmed if that's possible. And here, this is a huge percent. This is about 60% of the population of eosinophils expressing Lys6G because these are wild mice exposed to a lot of different, you know, parasites and things like that. So what do you look at in humans? The closest I can come up to is the idea that CD16 is expressed on a small population of eosinophils in humans. And the role of both of these is unclear. It's more of an identification. And then finally for cyclic F, there are two papers that describe cyclic F on neutrophils. I'm unsure what to make of some of the data, but this has been identified. And in humans, there is no evidence that neutrophils express cyclic 8. So I hope I confused you all because it is confusing to me. But it is more about paying attention and double checking which cell are you actually looking at. And this is where you go back to, you sort your cell and you do the H&E and you look at your cell because eosinophils hopefully always have those eosin staining granules. So here's my sidebar on mouse versus human. This is where I get to have some fun with controversy. So cyclic 8 is, you know, the human protein. Cyclic F is the mouse protein. They're functional paralogs. They were discovered by Bruce Buckner and Schleimer, in addition to other scientists in 2000. And they have spent a huge amount of energy characterizing the expression of these molecules on human and mouse. And so to point out, there are differences. Cyclic 8 in humans is expressed on eosinophils, mast cells, basophils. In mouse, it's eosinophils, alveolar macrophage, and some intestinal cells. So they are different there. They also have some similar ligands, I can't say it, those ligands there. And they have some different ligands. So in mouse, there's a MUC5B glycan that binds cyclic F. So they're similar yet different. And importantly, the antibody depletion, the antibody killing of human and mouse eosinophils is quite different. And this is where we get into a little bit of controversy. So in humans, if you use an antibody, you can kill eosinophils. In mouse, it's more confusing. So with humans with cyclic A, if you have the presence of IL-5 or other cytokines such as CSF, and you have an antibody, you can induce cell death. If you have antibodies such as the AK002, you can induce cell death also by ADCCs. So, but in mouse, you have cyclic F, and at this time, the commercial antibodies are, for example, IgG2A and IgG1, which are not necessarily as good at ADCC inducing death. The one antibody that's been shown to potentially induce some cell death is 9C7, which is not commercially available. So, you know, the question becomes, what do all those anti-cyclic F studies mean when these commercial antibodies are not really capable of inducing cell death? There's a question about, is it, you know, the epitope of some of these antibodies overlaps, or are we missing what we're depleting? Is it because these antibodies actually cause suppression of function rather than depletion? And I don't know the answer. It's just something to be aware of. So what about CCR3? Look at the clock. I'm going to totally run out of time. So human eosinophils, they express CCR3, but so do Th2 cells, basophils, and mast cells. Mouse does not have basophil, mast cell, or Th2 cell expression of CCR3 to any significant level. I think we often forget as well that CCR3 is expressed on stromal cells, so endothelial, muscle, epithelial. It's more extensive than just eosinophils. As far as the chemokines, eotaxins 1, 2, and 3 are made by humans, but mouse eotaxin 3 is a pseudogene. And just to remember that CCR3 can be inhibited by CXCL9, that was shown by Patty Fulkerson. I don't remember if this can occur with mouse eosinophils, and CCL11 can weakly bind some other chemokine receptors. I often see rantes mentioned as an eosinophil chemoattractant, and it turns out in mouse it's actually quite poor for eosinophils, so this is more of a human feature. And then it's just a reminder to pay attention to general mouse versus human situations. So in mice, CCL6 is now just recently found to be the paralog of CCL15 and 23. And then I know there's been a lot of research into the role of IL-18 in eosinophils, but mice do not make one of the main inhibitors of IL-18, which is IL-37. And on top of that, eosinophils do chemoattract to a lot of other stuff besides eotaxins. So complement, lipids. So, you know, if you find out your eotaxin isn't the answer, it could be lots of other things. I'm going to bypass this controversial topic, which is, I was just going to mention that CCL11 passes the blood-brain barrier, and it's associated with other neurological diseases and made by other cells. And as mentioned by Marc Rothenberg, eotaxin 1 is increased in patients treated with IL-5 biologics. So is this a concern if it can pass the blood-brain barrier? Is it a concern for long-term treatment with IL-5 for patients? You know, the neurological implications. And we did find eotaxin 1 upregulated in fill mice that are just shelf mice as well. So degranulation, just real quick, it's don't trust that this is actually happening in mouse eosinophils. It's actually exceedingly rare. And I had this picture, I just had to show it because it looks like an armadillo in our publication, where we show that on occasion in very pushed situations, which is this IL-5 human eotaxin mouse model, you can induce degranulation. And I'll bypass this because Ackerman already went over it. I just do want to comment that it was mentioned that EDN and ECP diverged in mice into ears. I still have colleagues trying to equate the two by buying proteins and saying that they're giving EDN to mice and things like that. If you search for ear 2, for example, on NCBI, do a blast search, it matches many other ears. So like here, for example. So it's not really appropriate to say that an M-ER2 is the equivalent of an EDN, for example. That's just my one opinion piece. Charcot-Leyden crystals, as mentioned, they're found in human and mouse. If you see a crystal, it is chitinase like 3 and 4, and this evidence of how these crystals work and that it's chitin here is quite an extensive work done by the Lambrecht group. And then finally, here I mentioned that human eosinophils degranulate in all forms and fashions. They respond to a huge number of triggers, and it has been shown that they do the piecemeal degranulation process and that there's preformed proteins in those eosinophils. With mice, it's becoming more and more recently shown that piecemeal degranulation can occur, but it's not entirely known what's inside those vesicles that are released and what induces it. It's less known if there's preformed granules. And these are just pretty pictures that show the sombrero formation in human eosinophils and then vesicle formation in mouse. And there are several labs now that have demonstrated this. So, because I don't want to run out of time, I'm just going to, so I want to go to Th1, so eosinophils and Th1. So we have shown that in mice, eosinophils can recruit T cells to tumors, they can induce Th1 activation, and in Th1 environment, they can kill tumors and they can present influenza antigen to CD8 T cells. And there's correlations of this occurring in humans. There's colocalization of T cells and eosinophils, and it's favorable for the stage, but it's not actually proven to date. I want to say, go read Ariel Munitz's review article. It's amazing. It has a lot more details on this. So eosinophils and immune suppression, our laboratory has shown that PD-L1 on eosinophils is critical to inhibiting CD8 Th1 activation and rejection. So lung transplants are better when eosinophils express PD-L1. There is a study with H. pylori that PD-L1 inhibits T cell activation, and more recently, eosinophil-derived TGF-beta was shown to promote Treg development. So is there PD-L1 on human eosinophils? And Ariel Munitz's group is asking, is PD-L1 a marker of a type 1 eosinophil? So yes, PD-L1. We took human eosinophils and activated them in type 1 conditions. They do express PD-L1. In humans, by others, the Lingblom papers show that a small population of CD16 high eosinophils suppress T cell proliferation and have the phenotype of elevated PD-L1. PD-L1 is upregulated in subsets of GI eosinophils and higher in patients with Crohn's. It's in some EoE patients. It's high on patients with COVID and related to COVID severity. And a recent paper showed that CD69 positive eosinophils related to COVID severity, and these express PD-L1. If you use checkpoint inhibitors that target PD-L1, there's evidence of an increase in eosinophilia, asthma, eosinophilic vasculitis, and a recent paper has shown that the eosinophil phenotype even changes in melanoma patients treated with checkpoint inhibitors. So the question is, should we think more about PD-L1 and T cells in humans? So my closing thoughts, since I'm running out of time, is mouse studies likely will continue to provide novel and otherwise unattainable insight into the potential of eosinophils in humans. Potential does not necessarily mean dictate necessity, yet may be evidence of a nuanced function, which when pressed or manipulated could have beneficial or negative consequences. And this is our group in Arizona, and I'd like to say thank you to everyone.
Thank you very much, Dr. Jacobsen.
Sorry, that was so hurried.
Talk is elegant and very detailed and shows how much information is out there, and you really have to study the details because there's a lot of fine differences and pathways that deserve a lot of attention. I think a lot of things you mentioned today probably could be the subject of another webinar, particularly the last information about the checkpoint inhibitors and PD1 and L1 and their involvement in eosinophil diseases in tumor and non-tumor states. So maybe we'll stay tuned for that going forward. There's not any questions that have come through from what I could see, so I'll just ask one briefly, but maybe it's just a point of clarification about the Siglec F in mice, because you implied that it works differently than maybe we expect, but as you know, there's a number of papers using anti-Siglec F to deplete eosinophils in mice, and then the consequences that have been examined. Are you saying that some of these should be called into question because the mechanisms may not be the simple depletion of eosinophils?
I do think that Bruce Bachner and his group really studied it thoroughly. And when you review the literature, it's hard to figure out, honestly, is what is going on? Why are some people showing depletion, even if they use different clones? I do think it needs to be studied more because there is a bit of controversy there as far as the different data, does it deplete or not? Because I also feel like it's one of those things that people say to each other behind public doors, oh yeah, that didn't work for me.
All right. Well, it's good to get the insight to these things.
Yeah, if I could just add briefly an additional comment to your question. In the papers that go to the trouble of actually looking to see by histology or other approaches if the eosinophils are gone, they're not. And both Siglec F and Siglec 8 get internalized when liganded by antibody. And many of these studies don't use a second marker like CCR3 that has not been targeted. And if you do, there's still plenty of CCR3 positive cells around. So I would just be very careful and make sure that you are or are not doing what you think you're doing with these antibodies.
It's terrific. It's great to see the expertise that the IES Society has. Thanks everyone for participating today. And this has been a great webinar. We look forward to logging in the future. Let's see, Andrew, if you could show some of the final slides that we have, because we are going to take a break for the summer. And we'll reconvene, I think, in September, and more information will be about that. Next slide, please. I want to thank our sponsors, which include Amgen, AstraZeneca, GSK, and Allakos. And the next slide, please. You can connect with us through Twitter and Facebook. Next, please. Please save these dates, July 11th to 15th, for the upcoming meeting next year. Looking forward to this. If you have any interest in recommending talks or you volunteer to speak, please let us know. The program committee is in formation stages now, looking for new ideas, and those are very welcome. Finally, thanks again to all the speakers and to Kate and Andrea at the IES Society for organizing this and administrating so nicely. Have a nice day and enjoy the summer break. Finally, yeah, that's a slide to remind us that there is an upcoming meeting this year in 2 weeks, which is the International Mast Cell and Basophil meeting, and there's going to be a joint session with the IES there. Welcome you also to log into their site to get more information. Bye-bye.